All right, welcome back, guys. We're going to go ahead and get started. Last time I did give you an assignment over pedigree charts. And the pedigree charts, I looked them over for many people, and there was a few confusing things regarding how to fill them out. So instead of giving you a big long explanation, I'm just going to go over that worksheet with you today so you can kind of see the key and how the genotypes should be labeled accordingly. So, just to give you an idea, in case you were not here, last time we did talk about the pedigree charts right here. We know that the squares represent males, the circles represent females, line between them, a bond or union of marriage or something like that in which they produce children, which is the next level down. And again, those individuals, they can have children of their own. So we're looking at several generations in this case, 1, 2, and 3 generations. So I am going to go to the chart here so you can see it. So this is what yours looked like, which was pedigree chart number 1. And you had to fill this one out. It said determine the genotypes if shading indicates brown eye color big B. The second one, of course, was determine the genotypes if shading indicates blue eyes. So you had a kind of a flip-flop of which one we were looking at, the dominant versus recessive. So let's just focus on number 1 for a moment here. All right, so in number 1, if we scroll on down, let's see the key, this is how it should have been filled out. And again, I saw some things that were not quite right on some of yours, and that's okay. So if you did have some mistakes, just maybe add a little private comment that said, I watched the video, I understand where I made the mistakes, and we'll go from there. I'll give you all your points. Okay? So as we look at this, it says determine the genotypes if the shading indicates brown eye color big B. So that means any of the individuals listed on here actually, or that have the shading, have at least one big B. Our goal is to figure out what is the second allele. Now you're thinking, why are there two alleles? Again, you get one from mom and one from dad. So we're looking at one particular trait in the individual, and we're looking at the two alleles that they possess, one from mom, one from dad. So all those that were shaded black should have at least one big B. And if you look across this example I provided you, they all have at least one. Now, all those that are shaded white, therefore they do not have the dominant allele. So therefore when we look at them, they do not have brown eyes. Therefore they must have the recessive allele, and let's say in this case it is blue. So we can go through and say, well, the only way that they don't have the dominant trait is if they have the recessive allele. And the only way to express the recessive allele is to have both of them. So if you notice, all of them that are shaded white in this case are little b, little b. Okay? So now, just looking over here, we have two parents that are going to be heterozygous. The question is, why are they heterozygous? Why aren't they big B, or something else? Well, if you look at the children, they had 1, 2, 3, and 4 children. 3 of which had brown eyes and 1 blue. Again, mom and dad had to donate an allele to the children. So one of these alleles had to come from dad, and one of them had to come from mom in this case. So that means both mom and dad had to have the recessive allele to donate, and therefore are heterozygous. Now, these two individuals right here, we look at them and we go, hey, why aren't they big B, little b like the mom and dad? It is possible that dad could have donated the big B and mom also the big B. Or dad donated the big B and mom the little b. So we don't really know what's going on with these two individuals as of right now. Okay. Now, if they had children later on, we could figure this out no problem. All right. So as we move on down, we go, what about this one? How do we know it's big B, little b? Again, they had children with this individual, and now we can see that the children, there are 3 of them that have blue eyes, which means the only way to get blue eyes is if both parents carry the recessive allele. In this case, they're both brown eyes, so therefore they must be carriers in this case. This one again is a question mark because it could be big B, or big B, little b. We don't know based upon the information we have. Okay? So as we look over here, we can see there's some other individuals that have the blue eyes and one that has the brown. We move up, we notice they had parents, one had blue, one had brown. But again, how do we know this one is heterozygous? The children. The children have both recessive alleles, so therefore mom also has to have it. This one right here is the cool one. Okay, so it's tricky. We know that this individual right here, this female, is the daughter of these two individuals. Some of you might have thought that they had two offspring, this son and daughter. If that is the case, that's incest, and that is not cool. This is not Arkansas, okay? So just kidding, Arkansas. You guys are cool. But as such, we know that one of the parents has the little b, but we don't know which one. We could take a guess, but we could be wrong in that guess. So when in doubt, you put a question mark, okay? Because there are some circumstances where you have enough information to guess, but it's not always going to be right, so you just put a question mark. All right, so the second one, as we look down here, This one's a little more elaborate, but it says determine the genotypes if the shading indicates blue eye color. So now those that are shaded, we are looking at the recessive trait, not the dominant trait, the recessive. So we flipped it up. So all of those that are shaded black in this case have little b, little b. As you can tell, every single one of them has little b, little b. All right. Now we can look at the ones that are shaded white. Therefore, if they don't have the recessive allele, they must possess one of the dominant alleles. So every single white one will have at least one big B. Our goal is to go back and figure out what is that second allele for each individual. Again, these two parents had one child, two recessive alleles, which means mom and dad both had to have the recessive allele. We go over here in this case. Mom had blue eyes, daughter had blue eyes. Dad must also donate the blue eye allele, therefore he's heterozygous. In this case, these two individuals had one offspring that had blue eyes, so therefore dad too must have donated a allele to make that child have blue eyes. Down here, two individuals that have brown eyes, but yet one blue-eyed child, which means they're both heterozygous. This one down here is the only one that is, in fact, a question mark. Okay, we know by looking at them, and because of the shading, they did not have blue eyes, so therefore they have brown eyes. So the question is, can we determine the second allele? And the answer is no. It could be big B, or big B, little b. In that case, we put a question mark. Okay, so again, if you miss some of these, you're looking back at your old one, you're like, ooh, I might have messed that up really bad. Just go back and say that you watched the video, you understand where your mistakes were. All right. Today, in place of your discussion board, you're going to get another one of these to hopefully master this content. Okay. So if you are confused on what to do, please go back and watch this segment of the video so you can actually figure it out. Okay. It's not real hard. You just got to use some logic and kind of work your way through it. It's kind of like a Sudoku puzzle. It's a lot of fun. Okay, once you get it figured out. Okay, so let's go ahead and jump back into the notes. Get started here. But real quick, is there any questions before we begin the notes? So if you have any questions, please post in the chat. It's always good to see you guys again. So check it out, the live chat there, and I'll be able to answer any questions you have. Okay, so we talked about this good old Punnett square, or not Punnett square, pedigree chart, sorry, and worked our way through this one. All right, so let's move on to the next segment. Inheritance patterns. Yay! So, we talked about what pedigree charts can be used for, but now let's talk about genetic abnormalities and disorders. So, how we inherit things. A genetic abnormality is an uncommon version of a trait that is not inherently life threatening. So, what that means is you can get a variation of a trait or an allele. So, for example, brown eyes was pretty much the standard trait that we had back in the day, way back in the day. Then, along the way, somebody mutated and had blue eyes. Cool, awesome. Are blue eyes life-threatening? And the answer is no. Okay, so you can have different traits and still be okay. So genetic abnormalities is just something that differs from the norm. Could there be somebody out there that one day that has orange eyes? Absolutely. It's an abnormality in the normal allele pool, but it's not life-threatening. Okay. A genetic disorder, on the other hand, usually causes medical problems that may be severe. A genetic disorder is often characterized by a specific set of symptoms, sometimes called a syndrome. And some of you, when you did your genetic mutations report, you found that sometimes they were referred to as syndromes, like Down syndrome. So, yes, some of these are really well studied and they have symptoms that cause problems later on for the individual or immediately after birth, whatever it may be. Okay, so as you look at this person's hands here, if it was just a issue with the fingers here, like they had an extra digit here, two thumbnails, is that necessarily life-threatening? No, not really. Is it a genetic abnormality? Yeah, it's not typical. You typically have 5 fingers on each hand. All right, you don't have typically 2 fingernails coming off your thumb. But is it going to kill them? Probably not. So there is a difference between abnormality and disorder. Make sure you note the difference. Okay, so there's different types of genetic variation. There are single gene variations. So an autosome, which is again chromosome 1 through 22, or a sex chromosome, 23rd chromosome, govern more than 6,000 genetic abnormalities. Whew, that's a lot. So again, many of these are what you reported on. Multiple gene variations, most human traits, including skin color, are polygenic and have some epigenetic contributions or causes. All right, so epigenetics is changes to the chromosome but not the DNA. So what does that mean? So epigenetics is talking about how your cells can turn DNA on or off. and that regulates what is expressed. So you may have genes in there that code for certain things, but they may never appear because they're not activated. So what do you mean? For example, all of your cells in all of your body have the exact same DNA, whether you get it from a lung cell, heart cell, brain cell, or stomach cell. They're all the same DNA. However, heart cells are going to continue to produce heart cells. Why don't they produce lung cells? Because there are many genes on that chromosome that have been turned off. So that's epigenetics. It's talking about how some things are turned on and some things are turned off. Now, could we take those heart cells and do some alterations to get them to turn into lung cells? It is possible. It is possible. They've been doing that with skin cells to turn them back into, or skin cells back into stem cells. So they have some of the processes down. Now, will we ever get there? I don't know. Okay, but that's epigenetics, which is different than eugenics. Eugenics is a totally different thing. That's what Hitler came up with his whole Aryan race thing. So don't get those two confused. Big difference. Epigenetics, good. All right. Eugenics, not so good. So epigenetics, again, just this diagram shows you how we actually can turn things on and off. A lot of the regulation of your genes is done by histones. Histones, if you recall, are those proteins that the DNA tightly wraps around to condense it down into that chromosome form. Sometimes there are these methyl groups that, once again, methylation is an epigenetic factor that is in some dietary sources and can tag DNA and activate or repress genes. So, again, there's more than just one thing out there. There's epigenetic factors, things like that. All sorts of crazy things out there that can turn genes on and off. Scientists have been playing with this to determine, hey, if this person has a genetic abnormality or disorder, is there a way that we could possibly administer something to their body to turn those genes off, which would be really, really cool for some individuals that are plagued by serious symptoms. So, epigenetics. So patterns of inheritance based on variations in a single gene. So this follows Mendelian patterns, and we're talking about Gregor Mendel. Gregor Mendel came up with the basics of genetics, the very basic understanding of how traits are passed on, dominant versus recessive, and things like that. So we're talking about autosomal dominant, which means, again, autosomes 1 through 22, and their dominant traits. They're autosomal recessives 1 through 22, X-linked, which is the sex-linked, okay, and this is on the 23rd chromosome, and also X-linked dominant. So we've got those. So based on variations in whole chromosome, changes in chromosome number, and changes in chromosome structure. So there's a big difference. If you get more chromosomes, so you typically have 46, if you get 47, that would may be Down syndrome. Changes in the chromosome structure, we're talking about breaking it, displacing segments of the chromosome altogether, deletions. There's also just massive mutations, whatever they may be. Right? As you look here, this is an example of Turner syndrome. Again, the individual has 45 chromosomes, not 46, and they only have one X chromosome. They are missing a Y or an X. Does this individual still exist? Yes, absolutely they do, but there is usually some symptoms or complications as well, but not necessarily are they super life-threatening. Okay, so patterns of inheritance for some genetic abnormalities and disorders. So abnormalities, you got things like achondroplasia, does not necessarily threaten their life, but it does cause dwarfism, shorter arms and legs. You got all sorts of them, and like I said, many of these were some of the genetic mutations that you guys looked up for your projects. The Marfan syndrome, missing connective tissue, polydactyl extra fingers, progeria was one. There is autosomal recessive inheritance patterns, albinism, doesn't necessarily threaten your life, but unless you try to go out in the sun a whole lot. But cystic fibrosis, though, is one that can be life-threatening. Definitely shortens the life expectancy of the individual. Friedreich's ataxia, sickle cell anemia, Tay-Sachs disease, all of these can be very, very bad too. X-linked or sex-linked recessive inheritance patterns. You've got red-green color blindness, which is the typical one that we talk about, and you just can't distinguish, colors very well. Doesn't make you necessarily life-threatening unless your whole world existed around, knowing which color is going to save your life. That would be bad, I guess. But hemophilia is bad. That's impaired blood clotting, which means if you get a cut or scrape, you could bleed to death. Sex-linked dominant, you've got the Fragile X syndrome, which is pretty bad, intellectual, emotional disability. So some pretty bad stuff on some of these. Changes in chromosome number. We talked about this. Down syndrome, Turner syndrome, Klinefelter's. You've got the extra X's or two Y's even. There's the Cryo-Duchat syndrome, which I know someone did theirs on that as well. The persistence in population. So if these things are so bad is the question, shouldn't they have weeded themselves out of the population by now? You're like, what do you mean? Like Darwin's theory on natural selection. If these genes are not favorable, they should have, these individuals died out, shouldn't have been selected for. They should have just stopped existing, right? All these symptoms and problems should have just eventually faded out to the pages of history. So why do they continue to persist is the question. So such mutations survive in populations for several reasons. Reintroduction by new mutations. So it is quite possible that an individual can have a mutation for, say, sickle cell trait, and then it's gone. But all of a sudden somebody has a new mutation and it brings it back. Mutations are random. They can happen at any time to anyone, and they can create new traits, just as this. Recessive alleles are masked in heterozygous, so you think it's gone, but it's just quietly hiding there in your DNA that you are unknowingly passing it on through many generations, and then with the right individuals, they both pass on the recessive trait, and bam, it winds up again. Heterozygous may have an advantage in a specific environment, such as sickle cell trait. Even though it causes some complications for the individual, it also grants them more resistance to malaria. So again, that's why it's persisting in certain countries or continents rather than just weeding itself out. Okay. Take home message. How do we study inheritance patterns in humans? Again, we can do this with pedigree charts, tracking generations through families. Abnormalities are rare versions of an inherited trait. A genetic disorder is an inherited condition that causes medical problems. Some human genetic traits are governed by a single gene and inherited in a Mendelian fashion. So, many others are influenced by multiple genes and epigenetics. And that again is controlling certain chromosome or genes on chromosomes. Okay? And we'll talk about the autosomal inheritance patterns another time. So I'm going to go ahead and leave it open here for you. If you have any questions, please post in the chat. Maybe it's about the worksheet, maybe it's about the lecture. Doesn't matter. Just go ahead and ask any questions that you have. Again, I'm always available for email. I do appreciate your emails. I've been very impressed with the work you guys have been doing. Your science dailies are looking really good. You guys are finding some really cool information. At first, there was a lot of it on COVID-19, but I see a lot of it is starting to shift to other things that you find interesting, which is good. Seen some really cool ones out there. So keep up the great work, guys. And hopefully you're all staying safe. All right, I've posted the quiz, the lecture quiz, and also your second pedigree chart. The second pedigree chart, I actually went through and made it a little user friendly for you. And let me see if I can pull it up. I robbed it from the biology class, but I made you guys a new one yourself here. So I know there was some problems last time regarding how to type on it and stuff like that. So I went through and I made some screenshots of the pedigree charts. So if you click on it, you can double click on it and it should open up a drawing box for you, and then you can add text boxes to label it if you want. Some people found some really cool ideas of how to use a pen, other things like that. It works great. So if that method worked for you, stick with it. But this is just another way to make it a little more user friendly for you. Okay? So that is pushed out to you. So again, if you have questions, let me know. Always watch the video and it should help you out. All right, I don't see any questions, so I'm going to turn you guys loose. Have a great day and enjoy the nice weather. And if you get bored, you can always watch The Adventures of Kentucky Coffee and Sheila Wallaby. There are 2 episodes out now. The third one will be coming soon. Thank you guys.